Hi, everyone, and welcome to this month's edition of the PAASTA Seminar Series. As the purpose of this seminar series is to foster connections and communication between all of us, we encourage everyone to ask lots of questions after the talk has ended. And as the presentation can contain published or unpublished data, we wish that any resulting content or conversation should not be shared without permission from the author. And if you are seeking interviews or statements for reporting, please contact the speaker to get express permission, as this is an informal presentation. And finally, we wish for this to be a safe and a positive space for knowledge sharing, so please abide to the PAASTA Code of Conduct. And with that, I am very excited to introduce Alex Burnett from Ghent University, who will be talking about their recent publication about ClassiCOL today. Alex, the floor is yours.
Thank you. Thanks for coming, everyone. So this is just a basic kind of walkthrough for how to use the ClassiCOL tool. It was just published this month, so if you want to find the publication, you can find it on the Journal of Proteome Research. Now, how this works, in essence, is that you should take your archaeological or forensic or otherwise bony sample, search it against a database as you normally would, be it with MaxQuant, Mascot, or whatever other search platform you use, but you should search it against a collagen database. You can then submit it to the pipeline that we've created. And what the IsoBlast algorithm first does is that it takes all of the peptides that are in your sample from your list of peptides from your search engine CSV, and it expands all of these peptides out into the total number of potential peptide candidates. It does this by calculating all of the possible isobaric peptides that there could be from each of your original peptides that match to the collagen DB. This then moves through into the ClassiCOL algorithm, whereby all of these potential peptide candidates are matched against the NCBI collagen taxonomy in a sort of bicluster. And eventually the output will show you a probabilistic sort of scoring, bearing in mind all of the possible taxa that could be in your sample. You can find all of the materials for the ClassiCOL algorithm and for the IsoBlast algorithm on Ian's GitHub. So it just looks a bit like this. There's not too much on there, but we are constantly updating it. So we do suggest that you use the most up-to-date version. And I'm going to show you today, using this most recent version, first a single species, single sample taxonomic determination, so a single bone that is sampled and analyzed. And then I'm going to talk through a bit what happens when you have multiple species that are represented within a single sample, and how that looks and how you can identify those. So the process is overall really simple. You just need your search engine outputs, be it a CSV from MaxQuant or Mascot, or if you don't use MaxQuant or Mascot, you can just submit a peptide list, and this is really simple. You just need your sequence and you need the modifications with the position. You need to download all of the ClassiCOL things from the GitHub, and that's all listed on the site really easily. So basically you just need the script, the requirements, the databases, and the distance matrix. And the distance matrix is really just to make it run a bit faster because it's pre-calculated some of the things that it needs in order to classify. And finally, you just need to execute the Anaconda prompt. So what does that look like? I'm going to show you some of the results. Hopefully you can see that. Can you see a web page?
We can still see your presentation.
Oh dear. I had a feeling something like this would happen. So what you'll essentially come out with, I'll just show you something different to start with. You'll first come out with a sunburst plot. So that sunburst is going to look something like this. And normally, as you can see here, you'll have a single species that pops out above all of the others. We also have a version of this where you can see not only the species that are present in the database and that are scored, but you can also see in gray or in black boxes all of the species that are in the NCBI taxonomy, but which aren't in our database. And that gives you a bit more insight into how you can discern and interpret your results. If, for example, the species that pops up, in this case, it's a rhino, you might not expect that to be at your site, but you might expect there to be a woolly rhino, for instance. We have a number of other outputs. So we also have a heat map, which is kind of informative because it shows you for all of the peptides and for all of the species, the scoring in that space. So it can be quite interesting to look at. And we also have a bar plot which shows you the unique peptides that are in your sample. I'm going to try and make sure that you can see my screen for this bit because it really is a bit difficult to explain without having it up. So I might, let's see. Sorry about this. I have a feeling if I exit presentative, it might. There we go.
Now we can see your everything.
Yes, and everything's just popped up, which isn't particularly useful either. There we go. So this is the sort of thing we're looking at here. So if we go across to this tab, this is that initial sunburst I mentioned, where you have the main outputs that are all scored in this nice kind of color-coded fashion with probability scores. So if you hover over, you can see, for example, here we have the overall score that's given to this taxon. You have the total number of peptides that contribute to that scoring, and you also have the number of peptides within that list, which can also be attributed to other species through the isoblast process. So how many peptides in this can also be attributed to another species because they're isobarics to other peptides that belong to that species. We also have, this is the one with missingness. So you can easily see here, for example, we have an outcome of Felis catus. But using this, we can also see that if in fact this would be Felis sylvestris, that's probably why it scores highly for Felis catus, if that doesn't make sense for your context. So here, for example, I might interpret this as wildcat, and if it's a more modern context, I might say that actually it probably is domestic cat. This is that bar plot. So this is a bit confusing to look at first, but what we have here is a clustering of all of the species that are outcomes in that sunburst. And you can see how far these overlap. It's interactive, which is quite useful. So I can see off the bat, there's not that much uniqueness for Canis lupus. There's not that much for, say, Felis catus. All of the Felis catus, the blue stripes there are all overlapping with other things there. So we can simplify our view, and you can keep simplifying it down until you can really interrogate those outcomes that were most prevalent in your data. And this is just kind of a way to visualize the peptides that are in your sample if you want to work with that a bit further. We also have, goodness, I can't get around my tabs around the screen sharing. There we go. You can also see here what the rescoring does. So we have a rescoring module in the ClassiCOL algorithm, whereby we take the original scoring and then we look only at the unique peptides, and then we rescore the outcomes only using the unique peptides from the top scoring taxa. And that's what produces this. So you can see for the species that are more likely, we tend to get an elevated score compared to the original score. And for those that are less likely, we see their score drop. And this gives us greater certainty in this sample, which was identified as being a hyena, most likely a cave hyena. There we go. We're going to go back to the PowerPoint now. And what we've got also from the outcome is you get a couple of CSVs. This one is the taxonomic results after rescoring. So for a single sample, that would be what this looks like. You have a list of all of the species that are in your sample, the rank that was given to that species, so that you can simplify this down easily to your top 8 candidates, your top 3 candidates, whatever you would like. And you can play around with that peptide data a little bit more. And it also shows you on the right-hand side, if you're using mascot in particular, your representative PSM title will be a unique identifier for the query. You can also submit a lot of files in a single folder. If you direct the algorithm to go to a particular folder, it will analyze everything in there. And then you'll come out with something like this. So this was only with 4 samples, but you can submit as many as you like, and they will all appear in this overview summary output file. And what this gives you, it tells you the name of the experiment. It tells you which taxonomic restriction you use. So for this, I restricted it to Mammalia. It tells you how many peptides are there for your first, second, and third species, which species they are, which protein, the peptide count, how many of those peptides again can be attributed to another species by isoblast, and how many of those peptides are unique for the species. This is automatically generated if you would submit a MaxQuant file where you've got your batch results. I don't use MaxQuant myself, so that's my understanding of how that works. And if you do Mascot, it will just be all of the individual files that you put in a folder. So what happens if, for example, you have an extinct species? So here we have that example I pulled up just a minute ago where we have a rhino output. Now, we know that rhino isn't necessarily what you would expect to find in Western Europe unless you're looking at a zoo context. And in this case, this is the sample of a coprolite from Skladina Cave in Belgium from the Middle Pleistocene. So we know that this most likely outcome is going to be a woolly rhino. And you can see again there in the rescoring that we really have an uptick and a fall away from that related species, which is horse, because horse and rhino are actually quite closely related. And this is where the rescoring really helps to discriminate those closely related species. So part of this, of course, will always be context, and part of it will be discrimination based on rescoring that helps to indicate. What happens if you have an extinct species which has multiple closely related species? Well, most of the time we see what you can see in the bottom right corner there, that we have all of the species that are related. So in this case, it's Elaphus maximus indicus and Loxodonta africana, because they're both close relatives of the woolly mammoth. So that's exactly what we see here. We see that those two closely related species have practically the same scoring, even after rescoring, and then everything else drastically diminishes. This is functionally, we call this a genetic mixture, because obviously the genes that feed into our protein sequences are going to be shared between close relatives, which produces this, this kind of equal matching. If you want to apply this on a sample from which you expect there to be multiple species, you go through basically the same process. The only modification this time from what I would do for a single species is you don't know what's in there necessarily, so you might not want to restrict your database, for example, just to mammalia. And you can use this for pottery, for soil, for calculus, for coprolites, for bags with mixed remains in them if you want to sample the dust, which we did ourselves. And the only other thing you might want to bear in mind here is that you might want to use a different type of FASTA, because we've got the collagen database, of course, but if you have an in-house database, for example, of dental calculus proteins that are similar between, or say milk proteins that are the same between different species, then you could replace the collagen database with a milk database, say beta-lactoglobulin. And the same goes for soil. If, for example, you're looking for particular pollen proteins and you have a number of ones from different plants, you can do that too. So this is kind of what that looks like when you have multiple species represented in a single sample. So again, we have something similar to what we see before with equal scores between different taxa. So you can see on the left-hand side here with this C16 coprolite, we have an indication of there being bear species in there. And in this case, it's probably a cave bear. We can also see that there's something of a signal for Felis catus, which again, we expect to be Felis sylvestris. And we also have some bovine signature popping up as well. And all of these have fairly equal scoring. Again, on the right-hand side, you can see a slightly simpler one of this, where I think here we have, and unfortunately, I can't really see it without the presenter mode. I believe this is woolly mammoth, that's the one. Woolly mammoth, boss species, and Felis catus again. We also wondered what happens if you merge samples. So this is kind of like an in silico mock-up of that single sample with multiple species in it. What happens when you merge individual samples on the data level? Well, on the left-hand side, you can see we have just woolly rhino samples. So this is multiple of them mixed together. And because they are closely related, we do still see quite a high score for Equus asinus, which is donkey. But we see that there is a higher score for woolly rhino species, or species closely related to the woolly rhino. If you introduce a single horse specimen into that mixture of data, what we see immediately is that that scoring becomes almost equal. So you do see representatively that the second sunburst there does show a mixture of species while the one on the left I would more easily interpret as being just a woolly rhino sample. This has been incredibly brief, and unfortunately, there was a bit more I wanted to show if my screen had been behaving. But if you have any questions, please feel free to fire away.
Thank you, Alex. This is very exciting. I'm just going to stop recording so that we can